Millicell cell culture inserts are designed to enhance the biological relevance of in vitro models. Traditional plastic plates restrict cells to a flat, 2D surface, limiting natural cellular polarity. The Millicell insert's permeable membrane allows cells to access media and nutrients both from the apical and basolateral directions, promoting improved growth and better physiological models. You can use Millicell inserts in applications such as barrier function assays, ADME toxicology screening, cellular migration and invasion assays, and co-culture systems. Our Millicell hanging inserts are available in 3 well formats, 5 pore sizes, and are either optically clear or translucent. You can refer to the user guide for more information on our full suite of inserts. Now let's learn more about how to use the Millicell hanging inserts. When you open a new hanging insert, peel the paper blister away from the plastic holder and use forceps to place the insert into a cell culture plate. We're demonstrating here with a 24-well hanging insert, one of the most commonly used formats. Note the marking on one of the phalanges or arms to help track the orientation of the insert. Ensure the phalanges are flat and not overlapping each other. The phalanges of the insert allows for easy access to the basolateral side without disturbing the insert. Inserts are tissue culture treated to support cell adhesion but can be coated with various matrices to help with cell adhesion. You can refer to the user guide for recommended media volumes and seeding densities. It is recommended that the media level of the apical and basolateral are even. Ensure all phalanges are flat to allow for the lid to close properly. Now, place the plate in the tissue culture incubator. If the membrane inserts are clear, you can monitor cell growth via microscopy. Here are some examples of data images taken on the Millicell DCI instrument. If inserts are translucent, utilize the Millicell ERS 3.0 for a non-invasive resistant measure of the monolayer. Depending on your assay, you may have to perform media exchanges during cellular growth. The apical assist rim facilitates media exchange without contact or risk of damaging the cellular monolayer. Come in at an angle with your pipette tip to the apical assist rim when performing media exchanges. Please refer to our Chapter 2 video for processing inserts for fluorescent imaging and H&E staining. For more information, please contact your account manager or visit our website.